Hi, in this video tutorial, I'm going to show you how you can analyze your immune repertoire data if you work with non-model organisms. For that, you would need a reference library, a custom one, and you can create it inside the Platforma. So here, I have a sample already added. This is a sample of IgH repertoire of a dog. So before running the MiXCR clonotyping block, as we usually do, I need a reference library, which is not built in MiXCR. I'm going to add a block, which is called MiXCR library builder. I'm going to add it to the project. Now I have to specify the name of the species, and in this case, it's going to be dog. It can be any name. Then they're going to be chains. And in this case, it's only going to be IgH chain, but if you have data for both IgH and IgL, or maybe T cell receptors, you can select many chains. And then you will specify the genes for each chain separately. And now the most important part is how do we provide the gene sequences? There are two options for each segment. First of all, we can use a built-in species. This option is good for example, humanized mice. So you can select V segments from mouse and then C gene from human, or vice versa. But in case when you have an entirely different genome, like a dog genome, we're going to use from FASTA file option. So I'm going to go and provide a FASTA file, which I have right here, with the sequences of my V genes for a dog. And then the covered region, I select V region because the sequences in my FASTA file start with framework 1. Most cases, this is going to be the option you're going to use unless your sequences start with UTR. And if it is the case, then you would select the V transcript. But I'm going to select the V region. Now I'm going to do the same for the J gene, also from FASTA file. And I have FASTA file for J genes right here. And these are minimal requirement parameters. But also I have sequences for the D genes. And I'm again going to select from FASTA file and add this FASTA file with D genes. That is all we need, and I'm going to click run. Now, and we can, while we're waiting, we can also look into the logs and we see how our genes, like each gene from our files, is being parsed and identified against a known species. The reference points are inferred, and the library is being created. Now, the important part here that is worth noticing is that the sequences in your FASTA file should be fully defined. Since it's a reference, you can't have any gaps or N nucleotides in the FASTA file for the reference. And here you see gene by gene, our library is being reconstructed. We can leave it for now, just wait, and while we're waiting, we can right away proceed and add another block, which we're going to use MiXCR clonotyping. So I'm going to add it to the project, and we still have to wait for the library to build, but right away here I can select a data set, which is going to be my custom dog data set. Then the preset can be either built in or from a file. In this case, I'm just going to use one of the built-in presets, for example, this one. And the important part is I have to select the chain, which I have in the reference. I can't select chains that do not present in the reference. In this case, I only have IG heavy. And the important part here, since we're using the custom reference, in advanced settings, first of all, under custom reference library, I'm going to select from library builder, custom library, and here is my library. I'm going to pick this one. And now, just for the purpose of this demonstration, since I don't want you to wait for the file to be fully processed, I'm going to select 10,000 reads here, which means that only this number of reads will be analyzed. It's a nice option if you just want to do like a dry run of your data to check whether everything works. Meanwhile, we see that MiXCR library builder finished working. I can click on this block and check that here I have the sequences of all of the genes from my file. This is the reference library that is going to be used. I'm going to go back to MiXCR clonotyping, click run, and now our sample will be aligned to the reference library provided. And since we selected only 10,000 reads, it's going to be very fast. That's pretty much it. So you see the alignment rate is more than 90%. So the reference library worked well. We see that we have only IgH in our data. And now, just as usual, you can proceed, for example, to the clonotype browser block and select your data. And there it is. Data has been aligned to the reference library that you provided. From here, you can use the output of MiXCR with all other downstream blocks, and it will work perfectly. That's it. Thank you for watching.